Okay, I think we're good to start. The recording should be running. And as I said, I'm sorry we're starting a little late. I had some technology issues, but we should be back up. If you do have any questions, these sessions are for you. So just let me know either in chat or just unmute and shout out at any stage. Otherwise, I'm just going to make a jump and make a start. And if others are joining us, they can do so in a little bit, and we'll just bring them up to speed. But I just don't want to keep you over time. So what I'm hoping to do today, we're at the week 7. I can't believe that things are going so fast. So what I'm hoping to do today is really focus on mentioning a couple of words about the lab that we'll be completing this week. It's a little different from the labs that you have completed so far, but this format will be something that we'll use a few times during this course. So I just want to kind of explain it through. I don't think it's anything super tricky, but so that no one feels that they're totally at a loss. The second thing that I do want to mention is about next week, when we're, can you believe, already moving on to the next set of exams. So just a quick walkthrough of what to expect. And of course, our main emphasis will be on reviewing the material from this week's chapters on the immune system and lymphatic system. It might seem like that's a lot of material to squeeze in 1 week, but really it's chapters that I think that often get overlooked in most of the A&P classes. So they are important, and I really want to spend some time that we at least discuss them rather than just skipping over what happens often in other courses. So we'll do the best. To be an expert is, of course, a different thing than what we'll be able to achieve here and now, but we'll do our best to have a basic understanding of both immune and lymphatic system. And with everything going on in the world at the time, I think that it's good to have basics of the immunology clear. So this week, there's actually going to be 4 pieces of assignment that I'm asking you to do. And the Learn Smart, quiz, and the practice anatomy, they're all assignment components that you're familiar with from the past. And they're all assignment components that you complete through the publisher's website at the Connect. The difference this week is going to be this immunology virtual lab, and we're going to complete that fully on the Canvas in terms of the grading component, and then in terms of the learning component, we'll actually go to an external website which is not your publisher. So just make sure that you're not going to be thinking that because it's not in the Canvas or it's not in the Connect that you wouldn't have to complete it. It still is an assignment that carries points towards your final grade. So make sure that you get it done. So it's something that's lab that gets completed in this BIO202 website. So you will find on the Canvas a link to this website. And this website walks you through various things about immunology, kind of like a lab process. What you will do as you're working through this lab, you will be filling in a worksheet that's available for you in a PDF format. So I would really encourage you to download it and print it, or you can just download it and fill it on your computer. Or if you don't want to print it, you can just answer to the questions on a normal sheet of paper, just write number 1 and then your answer. Or even if you want to do it on the Word, you can write the number 1 and fill in the answers. Most students find it easier to have that actual form in front of them. And what the form will look like, it's, I believe, 6 pages long. And it looks like what we're seeing here on this screen. So you will get a link of where to go to complete the lab. And then you have various questions of the lab as you're working. And I am looking for answers to all parts of the lab. I know that it's quite a few questions, but at the same time, you remember that the answers don't have to be super detailed. I'm just looking for an answer. It doesn't have to be an essay. So it's up to you how much detail you go into. And actually, it's rather quick to do if you just work through it at the same time as you're working through the lab. And once you have completed this lab on the biointeractive link and you have filled in the lab worksheet, you upload it back to Canvas for me to assign grades so I can see that you've completed it. Usually, as long as you've answered everything, everyone gets full marks for that. But if the answers are totally crazy or if there's a lot of missing, then it might not be exactly the full marks. Just to give you an idea also of the website, so you will find the link to the website in the learning module in the assignment. You click on there and it takes you there, and there you'll see a button, Launch Interactive Lab, and you'll complete that lab there. So I'm just including that so no one gets lost in terms of where to find the actual lab. Important part, like I said, is that make sure you don't forget to upload it then to Canvas. If you're having any issues with uploading it to Canvas, you can always send it to me as a message on Canvas or even as an email. But ideally, I would ask you to upload it to Canvas, but any issues, you let me know. We'll figure out a way to make it work. And I think that that's really all that I want to say in terms of this week's lab component. And let me just jump to another view. And the view that I want to show you is here. And I'm going to go to the student view on that. So again, because this class has been really, really good, but some other classes I've heard from my colleagues have found this lab a little bit more challenging. So I'm just going to also try to show this over here. So we go to this week's chapter material, which is the week 7 lymphatic and immune. And when you're completing the immunology virtual lab, you click in on there. You download the lab worksheet and it just automatically downloads it for you. It looks like we just saw, multiple pages long, 6 pages, I believe. Yep, the last page really is like half empty, so don't think that that's something that you have to worry too much about. And then you go to the link Biointeractive Lab. And there, to start it, use the button that I showed you, the Launch Interactive. And that's a lab where you then will be working through things as you need to complete this activity. Okay, we've had a look of that. So next thing for us to have a look of is going to be our this week's chapter materials key points. So let me pull that up. And we're going to start with the lymphatic system. Here we have it finally. That took me a while to get there, but we did get there. So what I want to do is to quickly highlight the key points of this chapter, what we need to know about the lymphatic system first, and then having a quick look of the immune system. So overall, if we think about all of the systems that we have in the body, and we've been focusing now on the cardiovascular system when we covered the heart, we covered the blood, the heart, and blood vessels. There is almost like a complementary system to that, which is going to be our lymphatic system. So if the blood vessels were like the tubing of the water system in a house, if you think of it this way, the drainage for what comes out of there would be then our lymphatic system. So it catches whatever happens to leak out from the cardiovascular system and returns it back to the cardiovascular system. In addition, it does a couple of other things, but that's kind of a good way to position your mind on why it matters. And of course, the clinical relevance is going to be when you go to the doctor. Doctor usually always, if we are thinking that there's an infection of some kind, we check out the lymph nodes. Neck is a very good place to notice them, but they're not definitely the only place where we have lymph nodes. If they are swollen, that is usually an indication that there is infection of some kind going on. And then that helps us to move ahead that, okay, now we need to find where the infection is taking place and how are we going to tackle it. So when we think of the parts of the lymphatic system, we notice that we have the lymphatic vessels, which are the tubing. Then we have the lymph, which is the liquid inside there. And then we have these lymph nodes that I already mentioned. The word lymph actually comes from Latin and refers to water. So that describes very well what lymph looks like. It's not like blood, so only certain elements of the blood manage to escape the cardiovascular system and therefore become interstitial fluid, and then that interstitial fluid gets picked up by the lymphatic system and becomes lymph. And if you think of the function of the lymph nodes, these are the sites where the lymph that's traveling through the system gets filtered and cleaned. And then there's a lot of immune system functions that take place there. So that's just to give us a quick review of what we're about to see. So we finished, I think, last week with me mentioning the hydrostatic pressure and osmotic pressure. These were the two pressures acting on blood vessels. So the hydrostatic pressure pushes the blood out from the blood vessel, whereas osmotic pressure is the force that pushes the blood back into the blood vessel. And usually these are fairly much balanced. And this happens, of course, probably the easiest at the site where we have capillaries, because that's where the wall is thinnest. And whatever is outside the capillaries, but not yet in the cells, is going to be then interstitial fluid. And the part of the blood that's able to escape these blood vessels as a result of the imbalance between hydrostatic and osmotic pressures is going to be plasma. And this, if we do the maths, we end up noticing that about 3 liters of plasma a day escapes in total from the cardiovascular system, from the vasculature. And this 3 liters is what we're dealing with. And what we need to put somewhere. If we are not going to put it somewhere, we are going to end up with then lymphedema, so the swelling of body because lymph building up to various parts. And what I wanted to speak here is fact that we need to use correct terms when we're discussing of these fluids, even though the fluids are very much alike. And they might be at parts exactly the same fluid. So we remember within the blood vessel, we talk about blood. But whatever leaks out of the blood vessel, that's our plasma, that becomes interstitial fluid. And the reason why it becomes interstitial fluid is that that's the name of the fluid that's not yet in cells, but it's not in the vasculature either. It's somewhere there in between. And interstitial fluid then gets picked up by the lymphatic system. And the moment it gets picked up by the lymphatic system, so the moment the interstitial fluid enters to the lymphatic vessel, it becomes lymph. But lymph and interstitial fluid have exactly same composition. So there's no difference in them, but we just use a different term. And I think that would be a very good exam question. I usually ask from the students that what makes, or when at what moment does interstitial fluid become lymph? And of course, we remember now that it is gonna be when it enters to the lymphatic system. Then interstitial fluid stops being interstitial fluid and now becomes the lymph. So that's, I guess, the big message that I'm trying to highlight here. And I think that we're ready to have a look of the connection between the lymphatic and immune system together. So you remember what I've drawn here is kind of the systemic loop of the cardiovascular system. So we see our heart, and from the heart, arteries leave. They travel all the way to the capillary network and then return through the veins back to the heart. And of course, we have arterioles and venules as part of this cycle as well. Well, we remember that most of this leaking from the cardiovascular system, leaking of the blood outside it, happened at the capillary level. And now it became interstitial fluid. And this interstitial fluid needed to be picked up by the lymphatic system. So let's have a look what we see when we add the lymphatic system here. Well, lymphatic system differs from the cardiovascular system in a way that it actually is not closed loop. It starts at the bottom of this diagram where it picks up the interstitial fluid, which becomes lymph, and then this lymph travels all the way close to the heart and gets dumped in back there to the cardiovascular system so that we're able to reuse these components. So that's the big difference. The direction of the lymph is only to one way, just as the direction of the blood in cardiovascular system is one way. But the big difference that lymphatic system is open system, open from one end. In terms of, if we think of the what kind of vessels we have in the lymphatic system, we have all the same stuff that we saw earlier on in terms of having the capillaries. These capillaries are what's the first part that picks up this lymph. And the only difference with these in comparison to what we see with the blood vessel capillaries is that these are even more permeable. So they're able to take up the interstitial fluid, which becomes lymph, but also various large molecules and particles. And that plays an important role in the function of the lymphatic system. So we pick up, in addition, something that can be so large that it couldn't be picked up by normal cardiovascular system from the capillaries to the blood. But if it's too large of a molecule, too large of a particle, the lymphatic system can pick it up. And then, as we saw earlier on here, the lymphatic system would simply just transport it up to close to the heart, where it gets dumped back to gets dumped to the cardiovascular system. So one time when we see this is, for example, when we're dealing with digestive system, and most of the nutrients, most of the energy from the digestive system gets picked up just fine. But large fats are often too large to be picked up by the cardiovascular system. So that's where the lymphatic system steps in. So if we want to list or think of the functions of the lymphatic system, and I think I have that coming up, but I'm just going to mention it now because it is something that's important. Lymphatic system serves basically three functions. It helps us to fight against diseases, so any infections, and that's where the white blood cells and lymph nodes play an important role. And we'll talk about all that in the second part of this week's material. In addition, lymphatic system helps to transport fluid. So we saw that balance when we did the math, that we don't want too much interstitial fluid to build up because then we would get an edema. So fluid transport is the second function. And the third function was this picking up large molecules, especially dietary fats, and getting these then to the bloodstream when they are too large in terms of the size of the molecules to enter normally back to the hold on a second. I'm just gonna pause quickly. I'm just gonna have to pause for a second. Why are you using Chrome? Sorry, just have it. Okay, apologies for that. Okay, apologies for that. I had a rodent and other critter exterminator trying to find their way back in, and they used different entrance than what normally. So I had to go and get them into the house. So apologies for that. And we're kind of left with the fact that I mentioned that the three functions of the lymphatic system were I'm just checking, hold on a second, were the fighting against diseases, dealing with this fluid balance, transporting fluids, and then dealing with the large dietary fats. And that takes us to have a look of the rest of the parts of the vessel. So we have capillaries, vessels, trunks, and ducts in the lymphatic system. And if you want to compare them to the cardiovascular system, they're very similar to what we saw with veins. So we end up finding, for example, valves in the lymphatic system that help us to make sure that the flow of the lymph is one way and one way only. And there's a couple of key locations that I want to mention in terms of talking what we might need to know. So here we have a view of the thoracic cavity, the posterior wall. So we see the ends of the ribs, and behind there we would have our vertebral column. We see right at the top the esophagus, and behind that trachea, or trachea first, more anteriorly, and behind that esophagus. So our windpipe, and behind the tube to run to the stomach. And we see our superior vena cava and inferior vena cava. So superior vena cava broad, all the blood from the upper parts of the body back to the heart, whereas inferior vena cava did the same for the lower parts of the body. And what we can see at the two structures at this diagram are going to be the thoracic duct. So that's the lymphatic duct that carries the lymph to the back towards the heart, and then structure known as cisterna chyli. That's this enlargement at the bottom of the thoracic duct, at the bottom part of the thoracic cavity. So that was our largest vessels and ducts. I want to show a couple of things about the trunks. So there's specific locations of the lymphatic vessels, and in this case, the trunks that we should have a look of. And we're going to start first here at the level of the lower part of the back. That's going to be our lumbar region. So, of course, these are named based on the body region that they serve. So our lumbar region, lumbar trunk for the lymphatic vessels, is located at the lower part of the back. Then we have our intestinal trunk, supplying the intestines. We have our intercostal trunk that serves between the ribs. So costal refers to the ribs, and inter refers to between. So between the ribs, we have our intercostal trunks. And then we have our bronchomediastinal. That's quite a mouthful. Broncho referring to the bronchi, and mediastinal referring to the mediastinum of the body. Trunk that serves that. We also have subclavian trunk, so below clavicles, such as just as we have subclavian artery and vein, and jugular trunk, which is located at the neck and throat region. So those are our trunks, and that brings us to then have a look of the ducts. And I've got a couple of ducts that I really want to highlight here. They are going to be larger. So we have our right lymphatic duct, which drains the right arm and right head and neck and thorax. And it empties to the right subclavian. And then we have our thoracic duct, which drains the remaining parts of the body and empties to the left subclavian. So subclavian is the blood vessel where the lymphatic system empties to. And we saw that quite nicely on that previous diagram. So now I want to talk about the lymphedema. Just quickly, I already mentioned that. We remember that edema was a swelling of some kind, and now because it's lymphedema, it's resulting from the lymph building up. And this would be typically because the normal return of the lymph is prevented one way or another. There can be, for example, tumors, or there can be issues with lymphatic vessels. They're not there or they're not working. And then for that reason, the lymph ends up building up to certain body part and we get this edema. So this may spontaneously improve over time as the other alternative lymphatic pathways or pathways of the lymphatic vessels enlarge over time. But this is not guaranteed by any means. And here's a couple of good pictures to just illustrate how can you spot that we have lymphedema. Well, usually the big thing there is that this would be only on one side of the body, so it would not be symmetrical on both sides of the body. And you can tell that it's not really fat buildup, it's simply fluid buildup from this swelling that we see. Well, next thing I think that would be to talk a little bit about what lymph is made of. What's the contents of the lymph? Well, lymph contains really the same composition as the tissue fluid or plasma or the interstitial fluid. It's just called different. So because it's escaped now out of the tissues or escaped out of the capillaries. So we find a lot of proteins. We also find, of course, a lot of water and white blood cells. One thing to notice here is that we should never have red blood cells in the lymph. So that would be a problem if we see that. And lymph is produced at the bone marrow and at the lymph nodes. So I think that that's all I want to say about these. And next thing would be to talk a little bit about the lymphoid cells. We're not going to go into great detail. The next chapter would look more into detail on that. So there's two types of cells. There's the immune system cells that are the kind of, if you think, proper. And then there's various supporting cells. And immune system cells are, of course, our lymphocytes. And we've seen lymphocytes earlier on when we discussed about the white blood cells. And the two that we're going to focus are the T cells, which mature at the thymus, and B cells, which mature at the bone marrow. So that's the simple explanation for their names. In addition, we do also have other cells such as macrophages and then supporting cells. And if we think of the functions of this lymphoid tissue, these are the surveillance points where the lymph gets filtered, but also a site if there is an infection where these cells that serve the immune system they get proliferated. So they are healthier, and then they grow numerous in numbers if we have an infection. And there's two types of lymphoid tissue. And we have this general tissue, which we can find really pretty much in everywhere in the body. And then we have these lymph nodes and various lymphatic organs. And I mentioned the lymph nodes when I explained the clinical relevance of this. So really about the lymph nodes, they are these kind of enlarged glands or enlarged parts of the lymphatic system. There's usually hundreds of those in one area. So they form kind of like a pearl necklace if you have a good imagination in appearance. And their function really was to be the site where the pathogens are detected, and then we respond to that by having these T cells and B cells and other white blood cells in more numerous numbers. They start to proliferate. So two functions for this: filter the lymph and look if there is something that shouldn't be, and respond to that. And there's again different regions for the lymph nodes, and they follow the body regions that we've seen before. So we have our cervical lymph nodes at the neck, and these would be what the doctor typically feels for, but that doesn't have to be the only site where we could check for lymph nodes. Are they swollen? Are they tender? Are they sensitive to touch? We also have axillary lymph nodes at the armpit. We have our supratrochlear, so above the trochlear notch. We have our inguinal, so at the groin area. For obvious reasons, we normally would not really test for those as the first option. We have our abdominal and our thoracic lymph node areas. And those are probably terms that I would know considering the exam. And this next one is just for my own entertainment, comparing the human and cat lymph nodes. We do see a lot of similarities, but there is also differences. So we do need to have vets. No human doctor can just move interchangeably from humans to animals. And the thing that I want to mention quickly is that we should have a look of these lymph nodes in a little bit more detail. So they are these small rounded blobs, kind of a bean-shaped. Some people describe them less than about 1 inch in size. And they do have a capsule that encloses them. And if we have a look of the different parts, the two parts that we find for lymph nodes are going to be the cortex, which was the outer part, the superficial part, and then the medulla. And here we have a very nice microscopic image where we can really see how the stain has picked up the different parts with different colors. And other thing that we end up seeing is that lymph nodes contain various sinuses. So these are sacs or cavities, just a general term that we can use for any organ or any tissue, really. So we find sinuses also in the lymph nodes. And here we have one lymph node that has been cut in half, cross-section of that. And we see that we've got a bunch of afferent vessels that bring the lymph into the lymph node. And then we have the lymph passing through the lymph node, and we have efferent vessels. So this is that take away the lymph from the lymph node. So be clear about the difference of afferent and efferent in terms of the lymph nodes. I'm trying to think what else could we say. We could say the clinical relevance about the swollen lymph nodes, because the reason why these lymph nodes swell is because now, as a response to infection, when we have detected that, the number of white blood cells has increased. And when you increase the amount of material in a structure, that will cause some sensitivity. So if it is painful or sore to touch the lymph node, that's usually an indication that there is infection of some kind going on. If there would be swollen lymph nodes, but patient wouldn't react in any way to touching of those, that would give us a reason to think that, whether there's something else involved there that's more serious. And last thing that we are going to do in terms of the part of the lymphatic system, we're just going to have a look through various lymphoid organs really quickly. I realized at the time I had to kind of start a little late and I had to run away. So I realized if you have to leave, just go ahead. But I'm going to go through these and then we will wrap up the second chapter real quick, or second part of the chapter real quick. But I want to start by talking a little bit about the spleen of the lymphoid organs or lymph organs. So this is approximately about a fist-sized structure on the left side of the abdominal cavity. It's actually, in fact, the largest lymphatic organ. You might remember the splenic artery when we discussed about when you reviewed the various main arteries and veins in the human body. And its functions really, that's our splenic artery coming in, and its functions really play a role in the immune surveillance and the response. So it cleans the blood of old or dead blood cells, platelets, and so on and so on. That's also a site where iron and platelets and a few other materials are stored. And in an embryo, the spleen is also the site for the production of the red blood cells. So what you'll end up seeing in newborns is that they do have a larger spleen because of this function of production of red blood cells has been taking place within the spleen. The next structure that I do want to talk about is gonna be our tonsils. So there's many, and tonsils are named based on their location. So palatine tonsils at the posterior oral cavity, lingual tonsils at the base of the tongue, pharyngeal tonsils at the posterior nasopharynx, and tubal tonsils at the auditory tubes. And I think that, again, their location is a clue of the function to kind of check if there is pathogens entering to the body that shouldn't be. And quite a few of us have had an inflammation of tonsils, which would be tonsillitis. And this can happen occasionally, and it is just part of the function, the purpose of the tonsils. But however, if this becomes chronic, you might have to get tonsillectomy, which is the removal of tonsils. That was quite common back in 80s and 90s as a procedure that we did for younger kids who had repeated tonsillitis cases. But we're now starting to question that a little bit, that tonsils do play an important role in the immune system, and maybe we should not remove them as easily as or be as inclined to remove them as we were in the past. So for that reason, it's not quite as common anymore. Anyone who has had their tonsils removed at an older age knows that it's a little bit more painful at that time. The next structure that I wanted to talk a little bit is going to be our Peyer's patches. They are in the small intestines and at the end parts of the small intestine wall, and function a little bit like tonsils, so destroy any bacteria, but also are the site for the memory lymphocytes to be housed. Couple of words about the appendix. That's the first part of the large intestines, and often I get asked by students that, well, you can get appendix inflammation, so why would you have this? What do you need it for? And the truth is that we don't really need it these days. It's an evolutionary remain. That's the most accepted theory at the moment. back when we ate food that was more plant-based, so the bacteria that's needed for digesting the plants are housed within the appendix, or warehoused within the appendix. And of course, if it gets inflamed, it does get removed. There's all sorts of things you can find from Chinese websites, including appendix stories and so on. The final one that I want to talk a little bit is going to be our thymus. And that's a tough one to find. You might remember from earlier on, we talked about that when you're an infant and in your puberty, we do still find it in younger individuals. But when you get older, it slowly gets replaced by the connective tissue and fat tissue. So it shrinks and it becomes harder and harder and harder to find. That doesn't mean that thymus stops working. It still works. It is the site of the T cell maturation production, but it's just not anatomically as visible structure. So that's all we need to talk about the lymphatic system. And like I said, I realized that we're running a little bit behind from where I wanted to be. So if you do have to jump out, I completely understand. What I'm going to mention next is a couple of words about the immune system. And we're really going to keep this brief. So I'm not expecting you to become experts of, but I do want to, for the sake of consistency, to mention few words that we have a basic appreciation of the immune system. So let's have a look of that. So immune system is really the defense system that protects us against diseases. And the reason why immune system is sometimes a little bit harder to cover in A&P classes is that it really doesn't have anatomical structures in terms of where it is or how it functions as much. It's much more physiology than what we saw with the lymphatic system. In fact, the immune system largely lives and is housed within the lymphatic system. So all of these cells and tissues and organs of these two chapters really work together, and the function is to protect against the foreign particles. So it's a very physiologically oriented system if we compare that. I'm trying to look. Next thing that I do want to mention is that what the immune system does, it protects us against different kinds of pathogens. And these pathogens are something that invades the body that shouldn't be there. And there's many different kinds. So when you are asked to explain what is a pathogen is really anything that causes diseases. It can be viruses, it can be bacteria, it can be parasite, it can be fungus, all these things that we see here. How these affect us and cause diseases is that they get into the human body and then multiply in numbers, which causes an infection. And I have few different kinds of diagrams of how you can classify these. If you're interested, I think I have already uploaded them to the learning module. They're not something that I would expect you to memorize. They're just for the good to know if you're passionate about this topic kind of a material. But if they're not there yet, I will make sure that they get uploaded after this session. There's two branches almost that I want to discuss, are two defense systems that we end up finding in the immune system. The first one that we're going to have a look of is going to be innate immunity. So this is very general responses, non-specific, and protects against many different kinds of pathogens, whereas this adaptive system is much what we kind of often think when we talk about immunity. It's much more specific and it targets specific antigens. So that's where we end up finding the lymphocytes, the white blood cells, playing important role in having this kind of a memory against of something that we've encountered before. For example, how vaccines would work, since that's something that's very relevant to this time. We're going to have a look of these both two in just a second, a little bit in more detail, but I do want to mention that there is a lot of overlap between the two. So I often find when we classify these things that it's good, but it does oversimplify the situation a little. So let's start with the innate defense system, which was our non-specific system. And this works through various levels of defense. So we can have physical barriers, such as skin or mucous membrane. If there's a break on either one, that usually is a direct route for a pathogen to get into the body. So we would be worried about that. And we typically can consider a comparison that these first-line defenses, physical barriers, are almost like the walls around a medieval castle. If the pathogen cannot get across that, we don't really have an issue to deal with or to be worried about. So they really physically stop as the first line that pathogen from getting in. The second line of defense is not so much physical, but more of a chemical response. So we have various different chemicals that would cause issues for the pathogens when they try to enter: sweat, stomach acid, tears, even urethra getting rinsed with the urine every now and then to wash away any pathogens that might be trying to get up and into the body through that. And the final line of defense is gonna be then our cell response, these cell reactions. If the two first lines of defense have failed, then we bring in various cellular responses. And we can end up seeing, and we'll cover a little bit about these different kinds of responses. Phagocytes which are kind of more of a white blood cells that eat foreign invaders. Something gets into the body, it gets caught by a phagocyte, it gets recognized and then caught. The phagocyte binds into it and takes it inside itself, and there it does break it into elements and components that are no longer a threat to us, and those can be eliminated. The other one that we might talk about a little bit is going to be our natural killer cells, so NK cells. So these are white blood cells that kill certain cells and promote inflammation. So you can consider them a little bit like as police guarding around and then acting if needed. And how they do that is by inducing a mechanism called apoptosis. And I'm sure you've heard about that before. Apoptosis was the programmed cell death. So that triggers the pathogen that's entered to actually commit programmed death. So it will not get in and cause issues. And this is very good picture of what this would look in reality. The third type of a cellular response is going to be inflammation. So I'm sure we've all heard about inflammations. Inflammations result in response to injury to body tissues, and inflammation serves many important purposes. You can get this injury, for example, from physical trauma or heat or chemical or infection. But what inflammation does, it stops or at least slows down the spreading of this pathogen and helps to remove these pathogens, but also helps with alerting rest of the immune system that we need to act on this. And the conditions of the inflammation, the fact that the site becomes warm, red, swollen, painful, sore, there might be loss of function. All this is ideal for the repair of this site. And what I want to do next here, I have pictures of 2 different kinds of inflammations, and I want to go through those signs and symptoms of inflammation with you. And I think that that would be a really good question in an exam as well. So what do we see in this picture? What can we tell about what happens in inflammation? Well, first of all, the site becomes very red. And that's because we have now a lot of blood cells flowing into this area. So that's why it changes in color, more reddish. There's a surge, a lot of blood cells flowing in there. The second thing is that it gets warm, the inflammation site. That's again, we think of the red blood cells, rest of the blood cells, white blood cells, platelets and plasma, all this rushing to this site. Usually blood is located more into the core of the body, so it transports also this heat with it to this site. As a result, because we have a lot of blood and blood cells flowing into this area, inflamed sites tend to swell, so they become swollen and they become sore to touch. Again, if you pack a lot of material between tissues, between the cells that are not normally there, that causes the physical stretching, which makes it also painful. And the final function is impaired function, so poor function of that site. And if you think about that, the pain and impaired function helps us to prevent from using and touching that site. We let it recover, we let it get better. And then this redness, heating, and swelling we could explain by the blood cells rushing to this area. The heat also serves another important function. A lot of these processes of recovery work better in a little bit warmer temperature than our typical tissue temperature. So these are local and flu-like symptoms that we end up finding on this site. I talk under the actual lecture video a little bit about the term pus, this kind of a combination of dead white blood cells, tissues, pathogens, and I explain why we use term purulent rather than anything else. So be careful when you write medical notes that you use correct terminology. The next one that I wanted to talk about was a little bit about the antimicrobial proteins. So what antimicrobial proteins do, they enhance the defense by attacking microorganisms and slowing down these microorganisms from reproducing. And interferons, which are often abbreviated as IFN, are very common ones, and they warn healthy neighboring cells and activate the immune system cells that there is some sort of a issue going on. And the last one that I really wanted to say a few words about in terms of the cellular responses on the innate defense system is going to be the fever. So what is fever? Fever is an abnormally high body temperature. And again, if we think of is fever good or bad? Well, let's think of the benefits. So fever actually inhibits the microbial growth, and there's also increased metabolism, so increased rate of chemical reactions within the body on that side, and increased phagocytosis, so this eating of the pathogens. So all of these things would be good in terms of dealing with an infection that we might have. And what we end up finding is that if you get sick, you rest at home, and we might give you medication to bring down your fever. That makes sense if your fever gets too high. Over 40 Celsius degrees at that temperature, a lot of your enzymes start to get destroyed, and that would be, of course, a concern for us. But if fever isn't that high, we tend to over-medicate people. We should actually probably let the fever run its course. If it doesn't get too high, and help with the recovery process. So that's something that we're looking into. And of course, some people do end up generating artificial fever in certain cultures, including my own, by having a sauna where an individual might go. And that would have, in theory, the same beneficial effects of a fever. Really, the second branch that we are left to look at is gonna be the adaptive defense system. This is very specific, as we saw. In here, we have met an pathogen before and we have built a response to that. And when we meet it again the next time, the actions against it are going to be much more quicker. So this is sometimes also described as acquired immunity, especially in UK and Europe. And I think that that makes a little bit more sense than hearing this term adaptive. So acquired is something that that the word really explains that you get infected once, you build the immune memory, immunity against it, and then when you meet it again, this pathogen, you have already a plan against it. So that's how you have acquired it. These are made of highly specialized cells and processes, and I'm probably not going to go into too much of a detail of this, but if we compare and contrast this innate immunity that we saw earlier on, and we spent a lot of time talking, and then this adaptive branch, we notice that we have very specific cells that we do want to focus here. And then we have two that overlap the two systems, the T cells and natural killer cells. In terms of the adaptive defenses, a couple of things that we want to note there is that these amplify the inflammatory responses. So inflammation, it almost becomes more powerful and activates also the complements which help. But the problem with this adaptive defense, because we can see that it works really well, very quickly to deal with these things, is that we need to have met this pathogen before. So we need to have been primed before. And this is achieved by having an infection, for example, meeting this pathogen naturally, or it could be, for example, a vaccination, small dose of this pathogen or inactivated part of this pathogen, or it could be also mother's antibodies that are passed to the baby through the breast milk and, of course, earlier on through the umbilical cord. So benefits of the adaptive immunity, since it is a little slower, is that because it is so specific, it recognizes the target immediately and acts on it, and it's not limited to the initial site where this encounter has taken place. So it acts throughout the entire system, entire body. And finally, this memory aspect, that a strong attack on the second exposure is possible because the antigen is already known to us. So what I want to say next really is that we have two branches within the adaptive immunity. One known as humoral immunity, which is uses antibodies to recognize the foreign substances, and these circulate free in the body fluids, and when they find an invader, they bind into those. And then we have our cell-mediated immunity branch, which are lymphocytes, so white blood cells in the lymphatic system, which can then act directly by killing the infected cell or calling other lymphocytes to deal with it and tagging them that, oh, these should be killed. These are problematic ones. Again, I have another nice handout that kind of summarizes the differences between T cells on the left-hand side, the blue circle, and B cells on the right-hand side, the pink circle. If you're really interested on this, that's available for you also at the canvas on this week's module. And really, I think that the last few things that I want to highlight is the terms antigen and antibody. I've used those few times now. I just want to make sure that we're clear of those. So antigen is the foreign substance that invades the body. So it's really kind of the target of this immune response. And it's something that's not normally in this host's body. Having an antigen present triggers an antibody to recognize and act on the pathogen. So these are immunoglobulins, often abbreviated as Igs, and these can be something that we test for. That do you have those? Here I have kind of more of a diagrammatic presentation. If you look, the antigen is the big blue foreign substance that shouldn't be there, and antibodies are these small Y-shaped structures which bind into the antigen to tag it that it shouldn't be there and let's act on it. This is what your textbook shows. There are actually many other shapes of the antibodies, so I just want to share that with you, that what you see is probably an oversimplification in the textbook. There's a lot more going on. And these work really like lock and key mechanisms, so they must be a match that we saw here. So antigen binding site is specific for a certain antibody. I think that other than that, really the cell types of the adaptive defenses I mentioned earlier on, the B cells and T cells, really the big thing there was to remember B cells matured at the bone marrow, T cells at the thymus. And then we have a third kind of cell type, which is the antigen presenting cells, APCs, often abbreviated. And these play an assistant role in showing an antigen to other immune system cells, so that we need to act on those. So let's have a look at the T and B cells a little bit. I think that we highlighted really nicely that B cells mature in bone marrow, T cells in thymus. These are the first or primary lymphoid organs, the bone marrow and thymus. And as they mature, they get tested for various functional accuracy. So there's a selection process. And then these go to the secondary lymphoid organs. And the secondary lymphoid organs, we'll have a look really quickly through just before wrapping up. We have our lymph nodes, we have our spleen, we have our tonsils, we have our appendix, and so on and so on. We discussed those earlier on this session. And then they end up circulating and eventually meeting an antigen that causes either the B or T cell to continue its development and then multiply in numbers and become activated. And this activation refers that it acts on the invader, so on this infection. And the memory cells are also created in response, so we can act quickly if we meet this same pathogen again. So I really think that this is all you need to know about T and B cells. And the last thing that I want to say, looks like our computer froze, is just a few words about these antigen presenting cells. So in terms of the antigen presenting cells, you can consider these as exactly what the name suggests. They meet an antigen, they cannot act on this antigen themselves, but they alert rest of the immune system that, hey, there is an antigen here, and this brings then the rest of the immune system to deal with those. So you can think of them as a little bit of helpers. So there's many different types of cells that can fit into here: dendritic, macrophages, and B cells. So really, to recap, we had our humoral immunity, which was the antibody causing it, and cell-mediated immunity, where no antibody is involved, but it's activated in response to an antigen. And last thing to kind of compare and contrast that I provided you was the comparison between primary and secondary immune responses. So in the primary immune response, that's the first encounter. We are exposed to something for the first time, and it takes about 3 to 6 days, during which you start to feel sick. It acts on number of days, if you think about it. The immunoglobulin levels are fairly low. Whereas the secondary immune response that we talked now later on, this is the same antigen that we're meeting for the second time. And we already have some sort of a plan for it. So the response is going to be very fast. It's actually within hours rather than days. And the response is also much more longer lasting than what we've seen before. And this is thanks to these memory cells. If we compare and contrast these, the primary immune response that we see, like we saw between 3 and 6 days, starting peak around the day 10 or so. But if we compare it to meeting this pathogen again for the second time, you'll see that secondary immune response is very, very fast. So in matter of hours. And it's much, much, much stronger as well. So this is all thanks to these memory cells. This next part, really, I just want to share with you that you can achieve the immunity naturally by encountering, being exposed to this virus or bacteria in your normal life, or it can be artificially acquired, for example, through a vaccine or the mother's antibodies. And really the big thing here to notice is that we can go into much more detail, but I'll leave that for your lecture video. And the last thing that I want to mention is that why would this then be relevant for us? Well, one time when this is especially relevant is when we have organ transplants. So we remove an organ from one body, place it into a recipient's body, and this is when the immune system becomes especially relevant. And transplants can be from your own body, so they can be from the same body. This is often the case in terms of blood or skin transfusions, or blood transfusions, skin transplants. It can be from a different individual as well. So this is the more traditional one, an individual of the same species. Or it can be even from different species, as for example with pig. Pigs are largely used for heart valves, monkeys as well. So most commonly we end up finding these allografts, which are transplants from one human to another, usually either living donor or dead donor. Dead donors are probably more common. So I guess that the good question with the transplants is that what are the most common organ transplants that we end up finding? Well, kidneys are really the most common ones. Oh, sorry, I jumped there. This is mainly because you can live with just one kidney. Liver as well, quite common because livers do have amazing regeneration abilities. So you can donate some of your liver and it grows back really quick. Hearts and lungs are not that easy, and we do need our own heart, we need our own lungs, but these are important organs to have, so that's why they're so high on the list. I guess that the next question that students ask me normally, how successful are these? How long of a time do they survive, or how do they survive over time? And we noticed that the transplants are initially very successful, but in about 50 years, or in about 10 years, about 50% of the patients end up rejecting the transplant that they have received. So that's kind of a sad news, but that's where we have to then consider, is a year extra time, 3 years, 5 years, 10 years worth it? So really how this relates to what we have discussed is the rejection of a transplant. And this comes down to the similarity of the tissue types, whether it's a blood types or antigens. And typically what we have to do when we do a transplant is that we have to knock out the patient's immune system so that they're able to tolerate that transplant and it doesn't get rejected. Well, this makes these individuals really vulnerable for other things, and really the leading cause of death for transplant patients is actually an infection, because their immune system had to be brought down in response to that. And really, I think that the last thing that I'm really going to mention just quickly is the immunodeficiencies. That's when we have impaired production or function of the immune cells. Some examples, Hodgkin's disease, that's when we have cancer of B cells, and we end up with depressed lymph nodes. AIDS, that's where the helper T cells activity is affected and often characterized by things like such as weight loss, night sweats, lymph node swelling, and usually it is the infections that end up being the killing cause of death. Really, the trouble is that we don't have really a treatment for this. We do have many antivirals that we can use as a preventive things, but we don't have or anti-exposure medication, but we don't have anything to reverse the situation. Many HIV/AIDS patients are able to live with undetectable viral load. Some others that I've listed here: rheumatoid arthritis, where the joints are destroyed, myasthenia gravis, where nerve muscle connections would be affected. I have MS listed there, so myelin, the insulation of the nerve cells, is affected here. Graves' disease, where the pancreatic cells are destroyed. Lupus, many organs are affected there, and then issues with the kidneys. So all of these are examples of autoimmune diseases. So in autoimmune diseases, the immune system attacks itself. So your body attacks itself when there shouldn't be an issue. And the question, I guess, is that how to treat these autoimmune diseases. Well, we can try to knock out or suppress your own immune system. We can give you anti-inflammatory drugs. And there is a lot of current research going on. So I'm pretty hopeful that autoimmune diseases are going to be managed better in the future. Other example that could be included into this chapter is also the hypersensitivities, where there's an over-the-top response to something that's normal. And good examples of those are allergies, and especially the anaphylaxis, where we might need an EpiPen to treat with the epinephrine patient. So this is all to do with those histamines and antihistamines that we talked earlier on. So that wraps up the review of the lecture material from this week. I know that we ran a little over. I will definitely keep that on my notes so that when the final grades are awarded, I know you've been here and you've been very, very patient. So I'll count that as a positive. And the only thing that we really have left is for me to remind you of the same things that I always remind you. I'm moving from one screen to another. Just bear with me. Here we are. I want to remind you that next week for the exam 2, the exam and practical are going to be available all week. I offer, so you can take it at any time that you wish. I offer a fairly, I think, comprehensive review guide, study guide, and we go through that study guide on Monday and Wednesday. On both days at 2 times, one starting at 10 o'clock and one starting at 8 o'clock. So I cover the content of the material of the chapters on heart, blood vessels, and lymphatic and immune system. Remember, in exam, you are only asked about the lecture video content. So those videos that I have recorded for you, there's going to be 50 matching multiple choice ordering questions like that. It is an open book. Tests, you can take it from home, so use your notes. I don't think that this time I have an extra credit question there, but I can see if I can come up with something. But at the moment, I don't have one scheduled, but or one added there. But like I said, use study guide, either attend to the review session or take part then by looking through the video recording of the review session if you feel that that's helpful. Usually I cover every single question from the exam on those review sessions. In terms of the practical, it is same as before, available for all week as well. And I really encourage you to review those. It covers the material from those practice anatomy practicals that you've done each week. But I also encourage you to make sure that you cover the practice version that I've got available before you take the actual practical, and I would recommend you to go through that multiple times because the questions are pulled from that. In the practice version, there might not be enough questions to cover everything, so go through it a few times. It is going to be, I looked through it in quite detail, various structures, also histology, and then vessels, whether it's blood vessels or lymphatic vessels, identifying those as well. 50 questions, which are the fill in the blank. Do not worry about the spelling like you saw last time. I will then manually fix it. You've got 60 minutes for that unless you have an accommodation plan. And again, an open book take-home test. So make good notes, have those available, and you will totally ace it. Other than that, I just want to remind you that I'm always happy to facilitate if you do want to retake or reattempt something that is an assignment and it didn't go your way. All you have to do is message me directly with very specific details what it is. And the tutoring that we have available, it's in a virtual setting for now. So using the Cranium Cafe, the link has been posted in various places, and then you just pick the tutor you want to speak with. But I'm also here, so do not hesitate to let me know. I've had quite a few appointments recently, so all you have to do is let me know when, and I'll make myself available. If I can't, I'll propose another time, and we can go through with anything that you might feel that is tricky from the material that we've covered. If you have any questions, this is a great time to do those. I know I kept you longer. I apologize for that. And that's really all that we have for this week. I know that usually students find this week's material in terms of the immune system and lymphatic system as quite a lot to get your head around, same as we had with the endocrine system. That's because both topics are so large that to fully understand which is really needed to feel super comfortable with it, we would need to spend probably a couple of years on these specific themes. So as long as you have a general idea, that's all that I'm asking for. If you have any questions, I'm here. If you don't have any questions, that's all I have for today. I really appreciate your good work on this course, and I really appreciate you sticking overtime. Like I said, I'll definitely count that on when I do the final grades as a very, very positive thing for you. So thank you for joining me. Let me know if you have questions. Otherwise, that's it for today. And on that note, we are going to wrap up the recording for this week's chapter. I will also have the afternoon session available. I always post these recordings, so they are available as well. And remember, for the next week, the big thing there is that be prepared for the exam, be prepared for the lab practical, and you'll do just fine.